A big hello to those interested in learning about experimental design, particularly for RNA sequencing experiment. This video will highlight the important parameters one needs to take into account before conducting an RNA-seq experiment. A good experimental design is necessary to maximize the power of the NGS study. Let's begin. A successful and robust experiment depends on a good design. So before sending your RNA samples for sequencing, or rather before even beginning the experiment, we need to ask certain basic questions. Why do we expect to see differentially expressed genes? What type of genes could be more expressed? What are the obvious sources of variability, the biological changes, and what factors or variables that can influence the outcome? The general goal of an RNA sequencing experiment is to find differentially expressed genes between two or more conditions. Let me give you an example. We could be interested in identifying the effect of a food supplement in a healthy group of individuals versus the effect of the food supplement in an anemic group of individuals or patients. Or let's take the second design. We could be interested in studying the differences in the gene expression between a wild-type cell line versus a cancer gene knockout cell line. Or we could be interested in studying the effect of an antibiotic in a bacteria versus a gene knockout bacteria. Like this, we have more and more examples in different fields of study. What are the general sources of variability in the data? There are two types, biological variability and technical variability. So the biological variability is the natural variation in the cell populations, organs, species, different individuals. Or it can also be the time points of an experiment cell cycle. Generally, the biological variation has been reported to be larger than the technical variation. But that does not imply that technical variability should be ignored in the experimental design of a study. High levels of noise in a technology must be considered during experimental design. There is not much to control in the case of biological variability since that is the natural differences in the biology. However, we can account for these differences by increasing the number of replicates. Anywhere between 4 to 10 replicates is a good option. Technical variability, such as the different flow cells used in the experiment, the different lanes in a given flow cell, library preparation batches, or different batch of reagents can all come under technical variability. Now, when you are having or when you're running the experiment with large number of samples, it's natural to spread them across different lanes. But we can account for this effect by multiplexing, introducing barcodes in the samples so that later they can be identified and demultiplexed. Batch effects can also occur by different days of isolation or library preparation. So we should make sure that we randomize this in order to account for it. So in an RNA-seq experiment, there's one important factor that we should think about. That is confounding variables. So generally we have something called as a treatment group in most of our experiments, and we have a control group in most of our experiments. And we generally, after say a given drug treatment, we harvest the tissues and the organs from both groups in order to be processed for the RNA sequencing experiments. While everything seems fine in this design, what if the researcher collects the different tissues and the organs at different time points? The treatment group is collected on one day and the control group is collected on another day. While this may not seem like a big problem, this kind of an effect or variation in the collection point is called a confounding variable, because now we do not know if the differences in the gene expression is only based on the drug treatment or it's also because of different time points. Like this, there are many confounding variables. It can be maybe the given diet for the mice, maybe the caging, the housing. In cases of cell lines, we have different passage numbers, different culture media. So one has to think, what are the other factors that can influence the main condition of interest? So what are the strategies to control for confounding variables? While we are talking about the confounding variables for the RNA-seq experiment, this can be used even in general experimental designs. So we have randomization, matching, restriction, statistical control. Firstly, why do we need to control for the confounding variables? To ensure the internal validity of your research, you must account for confounding variables. If you fail to do so, your results may not reflect the actual relationship between the variables that you're interested in. Randomization is, for example, while doing the RNA extraction, and you have a large group of, you have a large batch of samples, and you want to isolate the RNA from one batch on one day and another batch on another day. So sometimes we split those batches as treatment group on one day, control group on one day, but that is never advised. We should randomly assign the treatment in the control participants, or in this case the culture, to each group, and then carry forward with the isolation or sequencing experiment or even treatment. Matching. In this method, you can select a comparison group that matches with the treatment group. For example, you can match your subjects in your study based on age, based on gender. This will allow you to include a wider range of subjects. Next is restriction. For example, you can choose a constant sample collection time so that we are restricting this variation, and assigning also maybe the same laboratory technician to perform each library prep. Lastly, statistical control. After you've already collected your data, you can include the possible confounders in the regression model to control for the impact of the variable. Some of the practical considerations for RNA sequencing experiment would be the number of replicates. So generally when we prepare the libraries or send our samples to a sequencing center, we will be asked some basic questions. And one of that is replicates. So one has to choose the number of replicates based on the research question, based on the available funding. Anywhere between 4 to 10 would be good. Next is the coverage. So in general, coverage depends on your research, since the sequencing coverage is calculated based on the type of sequencing. For RNA-seq applications, coverage is calculated based on the transcriptome size. And for genome sequencing applications, coverage is calculated based on the genome size. But generally in RNA sequencing experiments, we are more interested in the read depth. Read depth is the number of reads per sample. It is used instead of coverage. So we always want a higher read depth to identify genes even with low expression. So in this example, it is the calculation for the whole genome sequencing, where we are using the Landau-Waterman equation, and C here stands for coverage, and it can be calculated by giving the number of reads times the read length divided by the genome size. For example, if you have sequenced for 5 million reads and it's a paired end sample for 100 base pairs and a genome size of 100 megabase pairs, we would get around 10x at genome level. So this is just an additional information for whole genome sequencing. How many reads? So for general RNA sequencing experiment for differential expression, anywhere between 5 to 25 million reads per sample is recommended. To find lowly expressed genes and alternative spliced gene expression, 30 to 60 million reads per sample can be given. For total transcriptome or finding new transcripts in the sample, somewhere between 100 to 200 million reads can be given. For smaller RNA, such as microRNA sequencing, fewer reads is sufficient. These are estimates and can, however, be confirmed with the sequencing center. Next, this is a general workflow for an RNA sequencing experiment, and this depicts the wet lab part of the RNA sequencing. So generally we have our tissues or samples or bacterial culture, and from which we are extracting the RNA using maybe customized kits or in-house lab protocols. So the RNA that we get is actually the total RNA. And in the total RNA, it's comprised of messenger RNA, ribosomal RNA, small non-coding RNA, long non-coding RNA. So it depends on the goal of the experiment. Now I'm talking about in general differential gene expression where we need the messenger RNA. So in cases where we need the messenger RNA, we will be asked by the sequencing facility center which type of either we choose a ribodepletion step or because we need to do this step called mRNA enrichment step. So where only the mRNA is focused among this total RNA. And to do that, there are three types of methods. So like I said, either the ribodepletion is done. As the name goes, the ribosomal RNA is removed. And then the poly-A selection. Now this type of selection is based on the poly-A tail in the mRNA. And so some kind of oligo-DT beads are attached and we're able to extract them. Next, a type of size selection is also done. So after this step is completed, we move on to the cDNA step. So yes, in the nanopore technology, even the direct RNA can be sequenced, but when we are going with the Illumina technology, it is generally reverse transcribed to the complementary DNA and that is sequenced. So with the cDNA, we are constructing the sequencing library and then we go for the PCR amplification and then the next step is the sequencing. So coming to the library, there is the unstranded library and there is a stranded library. With the unstranded library preparation protocol, there is no way to identify whether a read originated from the coding strand or its reverse complement. The non-directional protocols allow mapping of a read to a genomic location, but not the direction in which the RNA was transcribed. They are therefore used to count transcripts for known genes and are recommended for basic RNA-seq experiments. And the directional protocol, the stranded library preparation, preserves strand information and are useful for novel transcript discovery. Production of strand-specific RNA-seq libraries involves reverse transcription of RNA to generate the single-stranded cDNA, the complementary, synthesis of the second cDNA strand by PCR, and addition of SGS platform-specific adapters to the end of the cDNA molecules, either during the cDNA synthesis or afterwards via ligation. Hope this video was informative. Thank you.